So this leads to this notion that the DNA damage response activates the DNA sensor. We don't really know how the DNA gets into the cytoplasm or what obviously something we're now investigating. There seems to be a couple of steps missing here because we don't know how IRF3 influences the RNA turnover rate of RAY1 and another ligand, MALT1, that we've been studying. But in any case, these mRNAs are normally rapidly turned over, but the DNA damage response protects them. The other significance of this finding is how it ties into the role of this receptor ligand system in infections. And in infections, you can have indeed activation of various mediators along this pathway. For example, infections can lead to activation of the DNA sensor, or of course other sensors that activate IRF3. And indeed, some infections are likely to activate the DNA damage response, and recent studies show that in the case of HIV, that the VPR gene product, for example, activates ATR, and that can induce human NKG2D ligands in cells. So this shows a coordination of these different types of disease, a recognition of these different types of disease. Now, of course, there's a lot of other steps here in the production of these, and I just wanted to point out that the transcription of the gene was still mysterious, and what we have done more recently is looked at transcription via an indirect finding by a graduate student in the lab, Hae-Yoon Jung, and she made I think what was the surprising finding is that when we made primary cultures of fibroblasts, which don't express NKG2D ligands, under conditions where we stimulate them with lots of serum or defined growth factors, we see induction of RAY1. That actually is a sufficient signal with these primary cells in culture. And we see RAY1 not only RNA levels, but cell surface expression. This induction can be blocked by inhibitors of proliferation, shown here's no inhibitors, and then with various inhibitors, you see less induction of ligands. And you can actually starve the cells back and forth by adding serum or putting them in serum-free conditions, and that will toggle the RAY1 up and down accordingly in these relatively short-term cultures of fibroblasts. Many things happen when you introduce cells into culture, including, in some cases, activation of the DNA damage response or P19, but experiments that Hae-Yoon performed argue that those pathways are not central to this effect, or at least they're not the only cause of this effect, but that even when we inhibit these pathways, we still see such induction. So that led us to wonder whether proliferating normal cells in some cases upregulate NKG2D ligands, and we've only done limited work along these lines, but we were surprised to find that in very highly proliferative tissues like the E14 fetal brain, we could see really quite substantial expression of RAY1 on the surface of those cells. And indeed, since they dissociate readily without enzymes, we could perform flow cytometry and readily show expression at the cell surface in these E14 fetal brains. And then by E18, there's, according to the literature, the level of proliferation is much lessened, and indeed now the level of RAY1 expression decreases. I'll point out that at E14 there are really no lymphocytes in the mice, so we don't think this has any consequence for immune recognition of these early brain cells. But it does argue that hyperproliferation of cells in vivo can also lead to ligand induction. And I won't dwell on this, but Hae-Yoon went on to show that the induction here is actually at the transcriptional level by performing nuclear run-on assays, and arrested cells have very little transcription of the gene, and the proliferating cells have much higher levels. Now, so we began to think about signals associated with proliferation, and one of the ones we began to look at was the E2F family, and this shows a distribution of predicted E2F sites across the RAY1 gene, and there was many sort of clustered here near the promoter of the gene, which a graduate student, Ben Shung, had identified in the lab. And and of course, if we starve cells, I already mentioned that RAY1 levels go down, and of course E2F levels go down in parallel, as you'd expect, considering its role in proliferation. So we don't really have a complete set of results on this yet, but it is a role for E2F in transcription of RA1 is indeed suggested by the finding that E2F2 and actually E2F1 bind to the RA1 promoter in proliferating cells, but not in non-proliferating cells shown here. So here's the signal by chromatin immunoprecipitation of E2F2 in these cells, but not in the starved cells. This is another part of the gene well away from the promoter. And the E2F3 has so far failed to chip with the RA1 promoter. And this shows the IgG control. So this suggests, in fact, that E2F2 and 1 are associated with the RA1 promoter in these proliferating cells and likely play at least some role in that process, which we're now investigating. So this suggests that, taken together, that hyperproliferation here, perhaps by E2F and other factors, is inducing transcription of RA1, that the DNA damage response serves to stabilize the messenger RNA. In work I don't have time to tell you about, a graduate student in the lab, Tim Nice, showed that the proteins, at least some of the ligand proteins, are regulated at the turnover level by signals that may be associated with transformation, such as heat shock. So his work indicates that heat shock and actually UV light inhibit cellular ubiquitin ligases that otherwise would cause rapid turnover of the MALT1 protein, which is one of the ligands, so that if this is inhibited, you increase the protein levels markedly. And studies by several other groups have shown, and now our group, have shown that the expression of these ligands is also regulated at the cell surface because the protein is often shed as the result of matrix metalloproteinases that cleave it from the surface. The level of cleavage is often surprisingly high, leading to quite high levels of NKG2D ligands in the serum of cancer patients, which is believed, in fact, to cause inhibition of NKG2D positive cells systemically, suggesting this may have a role in tumor evasion, in fact. But whether this process is regulated or not, except for by expression of the relevant proteinases, we're not yet sure. So the implication, though, is that various signals associated with disease work together at different stages of biogenesis of these molecules to regulate the sensitivity of the cells to innate immunity. And so this suggests, and so here I'm speculating really, that possible barcode hypothesis, that is the idea that there are really several signals, different types of signals, each characteristic of cancer cells, that together are necessary for optimal ligand expression, and that these act at different stages of biogenesis of the molecules. But of course, there's also the redundancy aspect that some of these signals, depending on the cell, may be sufficient to increase expression in certain contexts as well. So I'd like to stop there and thank my various coworkers. I think I mentioned everyone along the way. So I'll finish up here then. Thank you very much.